So good afternoon to all. So I welcome you all for the guest lecture by Dr. Sendil Kumar from Department of Biotechnology, Mizoram University. So basically, he is alumni of this CPMB. He was here with us between 1997 to 98 to 99 as a research associate. Then he moved into a private college as a principal. Then again, he moved to the Central University to Mizoram. There he served as a dean for School of Life Sciences. Now he is a professor of biotechnology in Mizoram University. So to his credit, he is having, I think, more than 8 externally funded projects and also many peer-reviewed publications and more than 6 books. He authored, I think, more than 6 books, and also he was awarded with the many national and international fellowships. So with this, I invite him for the guest lecture on social genomics and diseases. And thank you all.
Thank you, Dr. Kokila, for the good introduction. So as Madam was mentioning, I had joined under Dr. Mohan Kumar in the year 1998 under the BPH project. Then SAR had left to US for a fellowship. Then I was working with Dr. Nagaraj. That time, Dr. Jnanam Madam was very young, and Madam used to always run in the department. And Dr. Arul was also there, and Dr. Kogila was PhD student at that time. So coming back to CPMB and TNAU is good remembrance, yeah, good feeling. And I thank the organizers for this opportunity as well. Being I just wanted to present some basic concepts, a general talk. It's not going to be a data-driven talk. It is a simple, compilation of ideas, basically. So I'll try to make this talk, as I mentioned, the general. So, like we all know that every evolutionary process leaves their distinct mark and trail on their genetic material. So basically, humans, we are also nearly 2 lakh years old. So our, Homo sapiens appeared on this earth approximately 2 lakh years old. And from there, different civilization, different societies actually has shaped our genes as well. From, as we all know, from foraging to post-industrial society. Today, it is a knowledge society or a data-driven society. So all these events have, made our genes modified and brought about changes that what we are all today. So this is again a very basic slide, how species evolve. So from the wild genotype as that in the time scale, as we see here, the polymorphisms or the changes accumulates within the individuals in different genes. Of course, we all know that protein coding genes do not alter as fast as the non-protein segment of the genome. But these changes brings about what the morphological or physiological or metabolic or other changes in the individuals. So there are 2 lineages that we see here. So over time, the wild type genotype produces 2 offsprings, and in the time evolution, the more number of progenies are produced, we find more number of changes. The basic difference between a mutation and a polymorphism is like polymorphism occurs at a higher frequency, mutation less than 1%, right, in a population. So we call that as a mutation. So these changes broadly defines what species are. So over time, if the lineage on the left-hand side cannot reproduce, the individuals on the left-hand side cannot reproduce with the individuals on the right-hand side, then we say that a new species has been created, right? So at every level, whether at a DNA or a RNA or a protein, we can monitor these changes, right? So the RNAs are also important, not only the genomics or the proteomics component, even the transcriptomes, right? Say, for example, understanding about the transcripts helps us to understand life. Like imagine all of us sitting in this room are traveling in a bus, and the bus meets with a small accident. All of us are human beings, right? So 99.9% or 8%, the genotype is the same. But we will find a lot of differences in the response to the incident, right? So some may be, shivering, some may be crying, some might have fainted, some, because of the accident, right? For some, nothing would have happened. They will be normal as such. So even though we all are same, 99.8%, the genotype is the same, but the response is different. So thousands of different types of RNA molecules are synthesized based on a stimulus. And all those mRNA molecules do not necessarily translate into a protein, but it will explain us the response. Like, for example, if you take a patient, uh, a cancer patient undergoing chemotherapy or radiotherapy, if we understand the mRNA concepts, we will know what is the response of the patient to the therapy, right? So at every level, we try to understand the basic biological functions. So, going back in the history, out of Africa hypothesis is widely accepted. The other end where the human genome is old, it's considered to be you know, the humans evolved from Africa and migrated to different parts of the world. That is called as old world, and this part of the world is called as new world, the North America and the South America. And the age of the human DNA here, basically, this phylogenetic tree was constructed based on the mitochondrial genome. So when we talk about the diversity of the humans, typically, we all know that the genotype interacts with the environment to produce a particular phenotype, and a large portion of the cultural transmission occurs from parents to children, from mother culture to the descendant cultures over the 2 lakh years, right? So natural selection plays a very important role here. We all, as a biologist, believe in Charles Darwin, and so the nature always selects the best of the best genes, right? And even today, many of the individuals face problems like infertility and things like that, which is actually not true, because any living organism will necessarily have the genes that they with which they need to reproduce, right? And that is the biological purpose of life, is to reproduce, right? The simple purpose of definition of life is to reproduce. So every individual can reproduce. It is only the thing that what is the natural selection mechanism that has happened behind the individual, whether they are able to reproduce or not. And we also know that genes can be always triggered. Genes are just like, the switch which we use for the light or the fan. If we want a gene to express, we can switch it on. If we want a gene to suppress, we can switch it off. So by simple manipulation of the genetic mechanisms, we can always, overcome even infertility. That is the basic concept of natural selection. So evolutionarily and population history-wise, they are responsible for the diversity, what we see here. Like as we saw in the previous slide, out of Africa hypothesis, due to environmental changes, due to other types of exposures that the individuals have undergone, we have adapted to our new environments. So, for example, dark people, they have sufficient amount of vitamin D, and white people, which where in Europe, more than half of the year there will be no sunlight, right? So they are white skinned. So the basic process of biological evolution is depending on the survival strategies, survival of the fittest. So over the generations. Classical example is antibiotic resistance, right? So which is also going to be a global problem, right? Even bacteria are more social uh, than the human beings, that they always transmit the resistant genes to their fellows very instantly, very easily, very freely, right? So they are more social than the humans themselves. So for example, if we go to the tribal populations who are not exposed to any antibiotic, with a single dose of antibiotic, they can overcome any type of infection. Whereas these days, when, for example, I remember when I was young, we need to take antibiotic for 3 days. Then it became 5 days. Then it became 7 days, right? So now it is more than even 10 or 11 days that we need to take antibiotic to overcome an infection, right? So these are all because of the social genomics that is lying behind the bacterial populations. Similarly, pesticide resistance, right? versus the organic farming, which was a cultural habit even for human beings. So pesticide resistance is also one of the major problem that we come across. So another classical example of biological evolution is elephants born without tusk, right? Because they don't find many natural enemies now in the forest. So they divert their energy in which they used to form the tusk to other necessity in their daily lives. So again, we all came across COVID, right? We all faced that. So the genetic variations in humans can lead to genetic predisposition or resistance to COVID, which was clearly visible across different populations in the world. US and Italian populations were more, at risk than few other populations. So it is also because of our social lifestyle and other habits. Similarly, the constant exposure to malaria, bacteria, and other viral diseases, right? Also, is a concept of social genomics. So I will now go back to little bit of natural selection. So as I told, my talk is generally a compilation of different ideas. Okay, so it's not going to be much of data. It is just to give a broad picture of social genomics, and we'll talk about some human diseases at a later stage. So the human intelligence is one of the classical example of natural selection. Like scientists did an experiment with this set of animal groups that what we see here, right? The experiment was just to take a simple piece of log and place it on another table. That was the experiment that was done across these animal groups. Like the modern prosimians, New World monkeys, Old World monkeys could not remember the task more than 3 or 4 times. So they need to be taught again what to do. Then they used to do the task, right? The task is to remove the piece of a log and place it on another table. Whereas the gibbons, orangutans, gorillas, and chimpanzees, they could remember up to 10 or 12 times. They used to do the same job, but again, they need to be taught. Whereas humans, we do a job and we never forget in our life, right? So the result of evolutionary pressure has been the selection for human intelligence. So further, they studied the brain size and the body size. They found that the horse, sheep, human, and the dog brains, right? They had compared it, and they found the human brain to be larger in size. Uh, it is proportionately bigger in primates. So human brain was 3 times larger than it would have been predicted. Because evolutionarily, based on the other organs in the human body, the human brain had a much faster evolution, right? 3 times it was larger than what it should have been. It is simply because we have three brains in one. We have a old reptilian brain, we have mammalian brain, and we have the new primate brain as well. It is one of the most rapid morphological changes in evolution. So again, how we are influenced by human nature and cultural diversity. Here is one classical example of Tibetan population. There was a publication on this. Mutation in the key gene allows Tibetans to thrive at high altitudes. The EGLN1 gene. So it was a hypoxic gene involved in the respiratory process. So they found that mutation has made this Tibetan population to adapt well, to survive, to do agriculture generation after generations, and thrive in the high altitudes where the oxygen level is very low, right? So whereas if we have to go there, many of us will feel suffocation. So that is one of the social genomic changes. Because of the lifestyle, because of the environmental changes, because of the adaptability, they have been adapted to that environment. So from outside, we are different due to the climate changes and differences in their culture. So how we define culture is behavioral traditions that are transmitted by social learning. At the population level, humans structure themselves into different cultures or ethno-linguistic groups, right? For example, Aryan race in the North Indian, Dravidian race in the South Indian, Mongoloid races in the northeast part of India also, and of course, different groups, right? The linguistic groups, there are 4 to 5 major groups for different world populations that have been classified. So hypothesis exists that the process of cultural evolution has played an active leading role in the evolution or selection of the genes. So many human genes that have been shown to be under recent or current selection are changing as a result of new environments created by cultural innovations. So like we saw in the first slide from the different geographic time zones of human evolution. From agriculture, we have moved into industrial and post-industrial societies. More genes evolved in humans because of the practice of agriculture. So humans also selectively transmit variants that they have learned to their offsprings because every organism wants their offsprings to lead a better life than what they did, right? Not only humans, every organism. So if we look into the co-evolution of culture and genes, the in the time scale given here, the genetic evolution happens. At the same time, cultural evolution also happens. And each of these interact at different levels at each time point. The influence of genes on culture through development is a classical part of evolutionary theory. Cultures have also impact on the gene pool. This occurs by culture modifying the selection acting on the genes, leading to different changes in the gene pool and also further evolution. So genes and culture are interlinked. Biological factors operate within a cultural context, and culture builds on biological foundation, right? So another classical example is cultures with a history of dairy farming, having cattle, and milk drinking have a much higher frequency of the genes responsible for lactose tolerance, the LCT genes, and its associated genes. Another classical example is malaria, right? So the culture and genetics are traditionally thought as two separate processes, but researchers are increasingly realizing that they are intimately connected. If we take the state of Africa, the yam farming was very predominant. That's a cultural practice of yam farming, which is just like a rice cultivation, having water stagnation in the yam fields. So because of that, there was an increase in the malarial population and mosquito population, and malaria was a bigger killer disease. But in the due course, humans have evolved resistance to malaria by forming the sickle cell, right? So natural selection may actually favor the sickle cell cells. So it is a gamble of life, right? Whether we die out of malaria or protect against malaria but have the sickle cell disease. So while it is sickle cell disease that's protective against malaria, it was uniquely human behavior or culture of yam farming that allowed the evolution to act. Another classical example is the Polynesian population, right? So the ancient Polynesians, they had a cultural practice of going on fish, you know, harvesting in the seas and oceans, and they used to travel for months together in boat. And whenever they used to reach an island, they used to get food for eating. Otherwise, they used to starve. So when they get a food, they used to take in bulk, store their energy for next 1 week or 10 days before they have their next meal. So that was their natural practice. But the modern Polynesians, right, because of fast boat and other cultural changes, from their own ancestors, they don't starve for food. So they have the problem of diabetes now because of their ancient cultural practice of having in bulk. So Polynesians settled in the island of Pacific, and they had a thrifty metabolism, which builds up fat deposit more quickly when food was available for them. And because of the change in the cultural practice, now that same behavior has led to the PPARGCA1 gene, leading to type 2 diabetes in the modern culture. Another example of cultural transmission is bottlenose dolphins, well known form for their foraging sponging. Right. So this behavior is almost exclusively transmitted from mothers to their offsprings through social learning. So it was, a professor from Carleton University, Jim Davis, who clearly put up a concept of if a behavior is present, it must be encouraged both by the genes or culture or both. So he had come out with this, principle. If there is a genetic predisposition for a behavior, and if there is a cultural support, the behavior is present in the individual. For example, let's take the case of smoking or alcohol, right? Inherently, if the genetic predisposition is there for an individual, if the ancestors was in the habit of drinking or smoking, and if there is a cultural support for them, then the behavior will be present in the individual. If there is a genetic predisposition and if there is no cultural support, then also the behavior will be present. If there is a genetic predisposition and if there is a cultural inhibition, right, if it is unlawful or the society doesn't accept that behavior, then the behavior may not be present or it is in struggle. If there is no genetic predisposition and if there is a cultural support, then also the behavior will be present. If there is no genetic predisposition and no cultural support, the behavior will not be present. No genetic predisposition, and if there is a cultural inhibition, the behavior will not be present. So, like that. Finally, genetic inhibition and cultural inhibition, the behavior will not be present. So, this gives the various combinations of the genes, the genetic makeup, and the cultural behavior. So, if we traditionally look into our own things also, like for example, some are vegetarians culturally from their ancestors, right? And the society also encourages to be a vegetarian, then they remain vegetarian, right? So, some may be traditionally non-vegetarians, right? Some may be fasting on few days, right? So that is because of cultural support that people starve on Mondays or Saturdays or things like that. So again, coming back to the example of the dairy farming, Northern Europeans drank more milk, where the Mediterraneans ate more cheese. Right. So over the years, the Northern Europeans evolved to be more likely able to digest lactose in their adulthood. So the problem what we have been facing till now is biologists are not trained in how culture can affect behavior, and anthropologists, sociologists, or cultural researchers are not trained in how genes work. Right. So it's now time that we combine both to see whether genes affect culture or culture affects the genes. Of course, it is a co-evolution process. So again, talking about diseases, like we have monogenic disorders, we have complex diseases. So monogenic disorders basically, is because of the Mendelian disease pattern. It is because of the single gene changes. Like we have cystic fibrosis, right? It is because of a CFTR gene that the disease comes into play. So if we look into the risk factors, it may be a dominant or a recessive pattern of inheritance. If the variant is there, the disease will be present. So if you compare it across the population, it will be all the same because it's a single gene. So if there is a mutation in the gene, definitely there is a genetic risk in the different families. Like here, there is a comparison of 4 families. Whereas if we go to a complex disorder, so multiple genes and environmental factors are involved. So the risk of this disease, like for example, breast cancer involving BRCA1 or 2 genes, among different families, the risk factors can be different. Right? It is because it is a multi-gene disorder. So it may be somatic, it may be germline. The variants can change, right? Even if there is a mutation, the individual or the families necessarily need not have the disease, right? So this is with respect to the genetic predisposition. So what biological factors can influence the disease? Of course, genetic variants, right? What type of mutations are present, and the genetic ancestry. Because if we want to know why a particular disease is prevalent in a particular population, Like for example, Northeast India, there is a high risk of cancers, right? The incidence of cancers is very, very high. So it is population specific. It is because linked with their ethnicity, it is linked with their ancestry, it is linked with their cultural behaviors. It can be biological also. So different environmental factors like exposure to toxins, cultural factors can also influence the diseases. So here is one example of the gene mutations in different populations. Like the mitochondrial genome is 16,500 base pair. Different genes are involved like ND1, ND2, CO1, CO2, CO3, ND4, ND5, cytochrome B. So these mutations are present in the cancer patients from the normal to the tumor samples or the cancer patients in different populations in a unique pattern, right? So for the same cancer, different mutations in different genes can be present, or for different cancers, different genes and different mutations can be present, and it is all population specific. So it is also linked with their cultures, habits, their ancestry, their ethnicity, and things like that. So the population associated diseases can be studied at uh, three different levels. One for prevention of the disease, like understanding the epidemiological and demographic factors. Uh, the second being detection, diagnosis, and treatment of illness. So even for understanding which drug will work in a better way, uh, which gene mutations are responsible for what type of diseases, and also for a better quality of life, right? So the genomic backgrounds of the populations have to be understood because different populations are different ethnically and also culturally. So the genomic databases are increasing in size and scope. More than 3,500 diseases and traits have been documented. Uh, as we prolong in the GWA studies, genome-wide association studies, more and more disease-related associations are also linked out. But the genomics is failing on diversity in the sense, uh, for example, if we take our own Indian population, not much studies have been done at the genomic level. Mostly it has been concentrated into Europeans, East Asians. In South Asia, India is not given importance, but other countries, African populations have been studied well, right? The Hispanic and Latino populations have been studied well. So, if to overcome this, because creating a database of variants in both healthy and diseased individuals is the need of the hour, uh, the DBT has come up with Genome India project, where 10,000 Indians have been sequenced now, the whole genomes, healthy individuals, different populations across the country. Of course, 10,000 is a very small number to cover a vast diversity of the Indian genomes. But this phase two also will cover more populations. So there, the entire whole genomes have been sequenced for understanding the disease-associated variants, to understand their allele frequencies, and also to come up with different diagnostic kits. So the impact on genomics on medicine and public health, there can be at different levels, starting from early diagnosis. Like if we have a good biomarker at the genetic level, even 5 years before a person can get cancer, those variations can be found, and the person can be, given a genetic counseling to understand, to overcome the risk, right? Uh, Similarly, preventive health, even in pharmacogenomics, also that's a very important field. So the one prime important thing is, like for example, um, uh, for breast and ovarian cancer, the BRCA1 gene susceptibility is found across different populations. So a lot of research has been done on these areas. So a detailed phenotype and geocoded data starting from the built environment, which gives the residential history of the person, including the environmental factors, the pollution, et cetera, the lifestyle, food habits, the socio-demographic factors, the questionnaires based on their lifestyle and food habits, the clinical parameters, and the genomic data will to integrate all this data for determinants of being healthy or determinants of being ill. So the common human variations are shared. Like for example, if we consider most sites that vary in the population are rare, most sites that vary in the individuals are common. We can try to understand what is the genetic makeup of the Asian population versus European and African, and we can come out with population-specific diagnostic tools or treatment decision tools, etc. So even in cancer statistics and cancer disparities, the based on the tumor types, men versus women cancers, nuclear gene involvement or mitochondrial gene involvement, highly mutated genes or frequently mutated genes, early onset disease or late onset disease, different pathways or the randomness of the disease, all can be understood if we understand the social genomics linking with the disease patterns. So the race and ancestry represent different but related factors. So there is always an interplay. Who we are, that means the social makeup versus the natural selection, lifestyle, food habits, our environmental factors. It's like kind of a one health concept, right? Combining social, environmental, and genetic factors, right? For a better understanding of life and life processes. Not only cancers but also diabetes, right? So diabetes is also prevalent across certain populations than the other populations across the world. So basically, this is one concept of pharmacogenomics. All patients with the same disease diagnosis, but there can be four different response, right? So one set of patient will have no benefit but toxicity, side effects of the drug. Right? Certain group of patients will have benefit of the treatment, but they will have side effects. Third group of patients will have benefit and no toxicity. So that's the best group of patients who are getting the goodness out of the treatment. The fourth group of patients will be no benefit and no toxicity, right? So here again, for the same disease, for the same drug, these are the four different classifications, right? So the best group of patients are they have benefit of the treatment and they have no toxicity, right? They have no side effects. So that is why personalized genomic medicines are into play now to understand the genetic makeup and then prescribe the drug based on their gene mutations. So many drugs are now available in the market. Like for example, even in the case of lung cancer, EGFR and KRAS gene, right? Mutations have specific treatment strategies. If a patient is having mutations in these two genes, they should be given a specific type of treatment. So, like we saw in the previous slide about cancer disparities, different types of cancers, there can be, treatments that can be prescribed based on specific gene mutations. So this slide tells us about the percentage of the patient population for which a particular drug in a class is ineffective. So cancer drugs, only 25% of the patients get benefit. 75% of the patients, the drug goes ineffective. Similarly, diabetes, 43% of the patients don't get benefit because of the metformin drug that is given uniformly to all the diabetic patients. So that is where we need to understand this genomics concepts. I already told you about the DBT Genome India project. So it is for identifying variation in the genome that is disease causing, and also to find out how individual genes play a role in health and disease. Because our genetic constitution plays a major part in determining our lifetime health. We are in the end of the talk. This is my last slide. So we attribute, this is one concept of Darwin, that it is not the strongest of the species that survives, nor the most intelligent that survives. It is the one that is most adaptable to change. Yeah. Thank you all. I hope I have not taken much of your time, your lunch time, basically. Yeah. Thank you. Yeah, yeah. So increasingly, sir, that even the non-coding part of the genomes are also now people look into it. because they code for small non-coding RNAs, the microRNA types, which also are involved in the disease process, the mechanism of the disease. So now many new concepts are emerging. People look into the non-coding gene also. They give a lot of importance, especially if it is in the splicing site regions. Then many research is going on with respect to different yeah. So like Sir is mentioning about Dr. Tangaraj and other researchers from CTFD and CCMB. They have linked with many of the genetic diseases with respect to the population specific. They have worked on Andaman tribes also with respect to diseases. They have worked on different communities, like Muslim communities having a specific syndromes. They have worked on the diabetic disease in specific Indian populations. So a lot of research is going on. They are also part of the DBT Genome India project, Dr. Tangaraj's group. Yes. Yes, yes. There are also groups who are resistant to this type of diseases. For example, in Mizoram, cancer is highly prevalent. In fact, according to the population-based cancer registry, Mizoram leads in both breast cancer in women, stomach cancer in men. It is because the population size, the entire state is having only 12 lakhs population, and they follow endogamy. Means they marry within themselves only. So they have very less choice of choosing the partners with a good genotype. And slowly over the years, because of their cultural habit of smoking, like 200, 250 years before also, they were living on hills. And anyone coming from the bottom of the hill, they will kill them with bow and arrow, even just 200 years before. And their cultural practice of farming is like to ward off the mosquitoes. Smoking is one of their cultural habit. Like father will light the cigarette, give it to mother. Mother will take a puff and give it to the daughter. Daughter will take a puff and give it to the son. That's their cultural habit to ward off the mosquitoes because they live in the forest. And because of these changes, like over the years, generation after generation, the mutations that are linked with tobacco consumption, the genes in their pathway are all fully mutated. And that's how they transmit it. They have no choice because they follow only endogamy, small population. So there is kind of a biomagnification of the mutations in their genomes, generation after generation. So it is one of the prime reasons why cancer is so prevalent. Like for example, we have found 21 base pair deletion in TP53 gene, which protects individuals against cancer. Similarly, novel mutations in tobacco pathway related genes, which have not been reported anywhere else in other human populations across the world. So there are unique communities and populations like in the Northeast also, which are either prone to cancers, but there are certain group of individuals, I mentioned about the antibiotic resistance. People who have not been exposed to antibiotics at all for many, many years. So if they have an infection, just with one tablet, they get cured in one day itself. So such type of medical cases are also there. So to understand their social life and social genomics will be very interesting to